Okay, hopefully everyone has had a chance to watch several videos on the central and peripheral nervous systems. We should have a clear understanding of the different structural features and a little bit about what each of them does. Your class notes use the figures from your textbook to go into the necessary details that you'll need to answer questions on quizzes and exams. So in this note set, our focus is going to be the general principles of sensory physiology. We'll look at the properties of the 5 sensory systems. I will cover the first one, which is vision, in this particular video. We'll talk about sensory receptors, perception of different stimuli, and the special sense organs. So our goal is to be able to identify the structures of the most common neuron, which is the multipolar neuron, shown here on the right. You need to know all of the structures and their function. A neuron's main function is to depolarize to threshold and send an electrical impulse in the form of an action potential. Then the Chapter 9 material helped us to understand and visualize the structures of the brain and spinal cord that send and receive information, again in the form of an action potential. So here we are in Chapter 10, where we can take a look at how we perceive the world around us. Our brain is constantly bombarded with signals telling us about the environment we are in. Figure 10.1 is a fun way of seeing how the brain can sometimes play tricks on us. But I want to go over some of the physiological details that so far these introductory videos already provided for you on Canvas haven't gotten into all of the details required for mastery of this chapter. The peripheral nervous system consists of 2 branches, afferent and efferent. Afferent meaning toward or incoming, and efferent meaning away or outgoing. These paths to and from the central nervous system, you may see or be asked to draw arrows toward or away from the central nervous system to indicate your understanding of the functional meaning of these terms. Afferent pathways send information in the form of action potentials about the external world to the central nervous system. Efferent pathways send information from the central nervous system to effectors. The peripheral nervous system is a sensory division that supports the central nervous system with incoming electrical information that generates our perception of the environment around us. Environmental information is detected by sensory receptors that respond only to certain types of stimuli. That is why you can't smell with your eyes or hear with your fingers. Visceral receptors detect stimuli from the body, and these include chemoreceptors located in major blood vessels in the head and neck region. They work to detect carbon dioxide levels, oxygen levels, and hydrogen ions that will control the pH of our blood. We'll talk more about that in Unit 4. We also have mechanoreceptors that monitor the degree of stretch and will fire when deformation occurs. One example is the tendons that attach muscle to bone. They send information about how heavy or how much force is needed to move a load. More on that in Unit 3 when we talk about muscles. We also have baroreceptors. Think of a barometer. Baroreceptors monitor your blood pressure. Proprioceptors provide a sense of balance and can be found in the vestibular structures in the inner ear. Nociceptors participate in the perception of pain. Please take a look at Table 10.1 in your textbook and also in your class notes. The brain utilizes these above types of visceral receptor information to regulate homeostasis, and we're not usually aware of these different types of perceptions. That's why they're called visceral. We'll talk more about autonomic responses in Chapter 11. The sensory receptors are structurally specialized and will only be able to respond to very particular forms of stimulus energy called a modality. Examples of modality include light, sound, pressure, temperature, and chemicals, like we tried on the neurons during lab number 3. An adequate stimulus is a receptor's response to its modality that causes a receptor neuron to reach threshold. Notice in both of these examples given here in figure 10.2, the stimulus is at the bottom of the diagram, and the action potential will move toward the central nervous system for evaluation, or perception in this case, along an afferent pathway, up towards the brain. Receptor stimuli and the 2-point discrimination test. Receptor physiology begins with an understanding of the key structures and their function. In order to send sensory information to the central nervous system for evaluation, the different modalities have to arrive at the appropriate sensory area in the brain. See figure 10.4. It is the job of the thalamus to project this electrical information along fibers toward the somatosensory cortex and sensory association areas in the brain. Structures at the dendritic endings of sensory neurons are specialized to respond to environmental or sensory stimulus energy. Activation results in changes in the membrane potential similar to an EPSP, which are always graded potentials. The receptor, also known as generator potentials, acts to trigger impulses toward the central nervous system along afferent pathways. Realization or perception of the stimulus occurs in the somatosensory cortex, shown in figure 10.4. Receptors can be a free nerve ending, which look like naked dendritic branches of an afferent neuron, or a cell closely associated with the peripheral ending of a neuron. These are compared for you side by side here in figure 10.5. Activation leads to the influx of sodium ions or calcium, depending on the sensory receptor, and these always lead to the depolarization activities in the cell. From figure 10.6, we can see the brain often integrates sensory information from different pathways using first-order and then second-order neurons to reach the thalamus, and then third-order neurons to project the inbound information onto the appropriate cortical area on the brain. Stimulus intensity is coded by the frequency of action potentials. Frequency coding shown here in figure 10.7. When we only consider the number of receptors activated, it's called population coding. In either case, a stronger stimulus leads to a larger receptor potential and will activate a larger number of receptors, and stronger stimuli, as we tested in lab number 3, will become more likely to fire the neuron to threshold and increase the rate of firing. Notice the graph at the top here in green. Those are action potentials coming along more frequently in time along the x-axis. Here we have a single sensory unit receiving weak stimulus on the left and much stronger stimulation on the right. These modified dendritic receptors become depolarized, and when the additive effects of EPSPs cause the receptor to reach threshold, an action potential is sent which codes for stimulus intensity with population coding. Remember, these graded potentials are proportional to the stimulus intensity. You can think of this as the action potential frequency being proportional to the amplitude of the receptor potential. In order for the brain to determine the location of the stimulus arriving from all over the body, indicating tactile, visual, or proprioceptive information, there must be what is called acuity. This is all based on the receptive fields highlighted here in blue, purple, and teal. Acuity depends on the size and number of receptive fields and the amount of overlap between them. Overlapping fields improves localization. Sensory neurons at the center of the stimulation area inhibit the more lateral neurons. Those are the ones on the right and left. In this example, we are using a blunt or rounded object to apply direct pressure to the skin. Lateral inhibition is a central nervous system process that sharpens sensation when a strong stimulus excites receptors from an exact location. Sensory neurons at the center of the stimulation area will inhibit the activity of lateral neurons by sending IPSPs, which are inhibitory. This process enhances contrast between strong and weak stimuli. In this case, the outer edges of a rounded object deform the mechanoreceptors less than the center of the object. Try this yourself. Close your eyes and press on your skin with the eraser of your pencil. And now try the same thing with the lead of your pencil. You can definitely tell the difference. 2-point discrimination test. Okay, this was normally carried out during lab, but we should be able to get the idea here by using figures 1010 and 1011. We use calipers in the lab to measure the distance between 2 points and touch the patient's skin each time while moving the 2 points closer and closer together until ultimately the patient cannot tell that 2 distinct points are touching them. The test is over when the patient says they can only feel 1 point, but you know there are really 2. The measured distance between 2 receptive fields is a measure of tactile acuity. Some receptive fields, like the ones on your face or fingertips, are more able to discern between 2 points than, say, if we did this experiment on your back or your shoulders. Let's transition to somatosensory sensations. Cutaneous sensations are sent to the central nervous system by mechanoreceptors. These receptors are buried in the dermal layers at different positions and respond to touch, pressure, heat, cold, and pain. There are both free and encapsulated nerve endings, shown in figure 1013. Free nerve endings are usually responsible for the sensation of temperature and pain. Cold receptors are going to be located in the upper part of the dermis, while perception of warmth comes from the receptors located deeper in the dermal layers. The encapsulated nerve endings mediate vibrations and constantly changing stimuli, so they tend to adapt quickly. These types include Meissner's and Pacinian corpuscles, which mediate pain. Free nerve endings that mediate touch or pressure include Ruffini endings and Merkel's discs. These are slow adapting and contribute to high-frequency action potentials sent up to the central nervous system. Adaptation of sensory receptors. The somatosensory system has the widest variety of receptor types due to the range of modalities it receives. Adaptation occurs when sensory receptors are subjected to a constant and unchanging stimulus. Think about your watch or your wedding band. After a while, you forget it's there. Receptor membranes become less responsive and receptor potentials decline in frequency or stop altogether. Receptors responding to touch and smell adapt quickly. Thank goodness. Let's talk for a minute about the perception of pain. Tissue damaging stimulation and pain is mediated by nociceptors. There are 3 types of cutaneous nociceptors. Mechanical, for example, stubbing your toe. Thermal, that detect intense heat. And there's polymodal. Poly meaning many, modes. That's going to be a mixture of both nociceptors and thermal and chemicals released from damaged tissues. That's why burns can be so painful for many days after injuries, because of the chemical release in the surrounding tissues. The body responds via an autonomic response. So the things that your body might experience during a painful episode would be increased blood pressure, increased heart rate, your body will release epinephrine, you'll have increased glucose blood circulating levels, you may even have some perspiration or sweating. The emotional response can include anxiety or fear and a reflexive withdrawal from painful stimuli. There are 2 types of pain, fast pain and slow pain. Fast pain is perceived like a knife or a sharp pain. Slow pain feels more diffuse, less localized. You can see figure 1016 for a map of referred pain. The second order neurons that ascend to the thalamus via the spinal thalamic tract can provide discrimination of the perception of pain. The spinal thalamic tract transmits information from both thermoreceptors and nociceptors, but the perception of pain will involve the neurotransmitter called substance P. The emotions evoked from painful experiences involve the limbic system. Notice here the first order ascending neuron is in the left, and the opposite side of the central nervous system is receiving and processing the information. This kind of crossover is called decussation. Now let's look at the first in our series of special senses. Let's start with the eyes and vision. Your assigned reading for eyes and vision will be pages 269 through 284. As you may have already learned from previously posted videos here on your Canvas page, the anatomy of the human eye is slightly irregular and hollow, forming a globe-like shape. There are anterior and posterior poles. The anterior aspect is where the light modality will enter through the pupil and becomes focused on the posterior aspect or retina, where the sensory tunic holds the photoreceptors called cones and rods. The outer wall of the eye is composed of the white sclera. It is fibrous and less sensitive to touch and pressure. The middle tunic or layer is called the choroid. It's vascular and shown here in a dark purple band that goes around the back of the eye. The internal cavity of the eye is fluid-filled. The anterior chamber is filled with aqueous humor, while the large interior of the eye contains vitreous humor. Humor is a Latin term for fluid. The lens is what separates the internal cavity into anterior and posterior segments. In this anterior segment, both cornea and lens are convex in shape. Those surfaces are transparent, flexible, and they lack vascularity. These structures allow for precise focusing of light onto the retina, which is composed of 3 layers, shown in figure 10.2.9. The human eye can only detect certain ranges of light. It's a very narrow light spectrum, basically just the colors of the rainbow. You can remember them in order from the acronym ROY-G-BIV: red, orange, yellow, green, blue, indigo, and violet. Visible wavelengths of color fall between 750 and 350 nanometers. Beyond these ranges are invisible light forms to us. We cannot see UV light, but we've all experienced a sunburn. On the other side is the infrared waves. And you may have seen the military use night vision goggles in order to see heat in this part of the spectrum. In order for us to see the light reflected back to us from observed objects, the light waves must converge on a single point on the retina in order for us to see them in focus, or we may require corrective lenses to help us focus. Convex surfaces allow light to converge on a single focal point. Vision that uses the center of the visual field and a place on the retina called the fovea is where visual acuity is at its highest. The fovea is responsible for sharp central vision, which is necessary in humans for reading, driving, and any activity where visual detail is required. There is a high concentration of cones located here. Accommodation. This is a term used to describe the ability of the lens to become more round and able to focus the light coming from objects near to our eyes. Accommodation is also something we did when we tested cranial nerve 3, the ocular motor nerve, in lab 3. Here we see the pupil. They're able to accommodate to dark and light environments. And starting here on the left with the normal sized pupil, bright light or a parasympathetic sympathetic stimulus can cause the pupil to shrink, which protects the retinal layer in the in the back of the eye. Our eyes can also adjust to dim light by dilation, so the pupil is going to get bigger, and that's going to allow more light to come into the eye. When we look at the delicate retinal layer, it's made up of 3 layers, consisting of a pigmented layer, which is the outer layer and absorbs packets of light that enter the eye and prevents things like scatter, which will cause distortion and lack of visual acuity. Here is the cones and rods. They're located in this layer. The middle is filled with neurons and contains bipolar neurons along the amacrine and horizontal cells that modulate communication between all the cells in the retina, and then the inner layer of neurons that are called ganglion cells. The name ganglia refers to these large bump-like structures drawn here in yellow on the left where the light waves are coming in. Light that passes through the retina gets absorbed by the dark choroid layer, and some of this light stimulates the photoreceptors, which in turn activate action potentials. The phototransduction process of converting light energy into electrical signals, we utilize the cones and rods for both light and dark vision. They contain numerous sodium channels in their plasma membranes on the outer segment. Rods shown here on the left respond to dim light and are helpful with peripheral vision. And cones here on the right respond only in bright light conditions and provide high acuity color vision, and they're located in high concentrations near the central fovea. During signal transduction of light modalities, the cones and rods will respond independently. In dark conditions, notice here the photoreceptor releases its chemical transmitter, but when light is present, it gets absorbed by the photopigment. Examples of photopigments would be opsin or rhodopsin. That ultimately is going to shut down the voltage-operated calcium channels and decreases the release of neurotransmitters. You can see this by comparing these two diagrams in figure 1033. Notice the numbered steps. When we take a look at the neural pathway, we see decussation again, where the pathways cross over. The right half of our visual field projects onto the left half of the retina, and vice versa. The receptive field is the part of visual field that affects the acuity of a particular ganglion cell. Its activity will be modulated, and these neurons form the optic nerve, which is cranial nerve 2. Each eye has an optic nerve that exits at the optic disc. And this is also called your blind spot since there are no cones or rods located here. Cranial nerve 2 combines at the base of the brain, forming the optic chiasma, and is another site of decussation. Left lateral geniculate nucleus receives input from the right half of the visual field of both eyes, and the right lateral geniculate receives input from the left half of the visual field.